The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, EPA Regulatory and Testing Guidelines for Residual Efficacy, How to Make Antimicrobial Product Claims. Now, if you missed one of our webinars or would simply like to refer to one, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars by liking Nelson Labs on Facebook or following on Twitter or LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There, you'll find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our experts and advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for healthcare, consumer, medtech, and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and consumer industries. The experts at Nelson know that every test matters and requires solution to complex problems to improve patient outcomes and minimize client risk. And now let's get started. Today we're joined by Grady Wertman. Grady has worked at Nelson Labs Bozeman for over 8 years. Grady holds undergraduate degrees in cellular biology, neuroscience, and microbiology from Montana State University, Bozeman. He joined the team in December of 2013 as a laboratory technician, but has since then become an integral study director at the facility and one of our leading subject experts on EPA testing and regulations. We do encourage your questions and submit them at any time in the question box. Grady will try to answer as many as he can in the last 10 to 15 minutes of the webinar. Now, beginning today's presentation, I turn the time over to Grady.
Thank you, Daniel. So today we are discussing the recent regulation updates to the EPA claims for residual efficacy. We're going to be focusing on what's required for these claims and what methods and SOPs from the EPA we will be following to perform these tests. So as we're proceeding through this webinar, there's some terms that are going to be thrown around that in the grand scheme of things have very similar meanings. But when we are applying this to the EPA, the EPA is looking for very specific definitions. So antimicrobials are just the general substances that kill microorganisms. Disinfectants, again, a chemical substance used to destroy microorganisms. In self-sanitizing, the ability of an antimicrobial product to reduce populations of bacteria residually. And so, but you know, it's kind of, to put it in a nicer term, all E. coli are bacteria, but not all bacteria are E. coli. So what we're looking at here is the terms seem very generic, but they are very specific when it comes down to EPA claims. So a little history on the residual efficacy here. Back in 2020, we were in the throes of the COVID-19 pandemic, and the EPA had received a lot of requests from stakeholders to provide some sort of guidance on residual efficacy for their product. You know, they wanted to show and be able to say that their product continues to reduce microbial populations, whether viral or bacterial, over the course of a set amount of time. So what the EPA had done is they released the interim method for evaluating the efficacy of antimicrobial surface coatings. And so that method is kind of a conglomeration of what we'll go through in this, whereas everything was packed into one method and then it was pulled out into individual components over the next couple of years. So for the next couple of years, the EPA received public comment from laboratories, from consultants, and other stakeholders, and Then they also performed internal testing to gather data on these methods and determine what aspects needed to be tested and how can we prove that the products are efficacious for 1 week, 3 weeks, 5 years, 10 years, undefined length amount of time. And so what they did is in October of 2022, this year, they actually released 3 guidance documents that clarified all of that and wrapped it up in a nice little package. And so what they're planned to do is to incorporate these into the relevant OCSPP Series 810 test guidelines once the next cycle comes around for updating those. But what they really introduced were 2 new SOPs and a guidance webpage. And so their guidance webpage really just lays out everything that you need for this, including which methods to use, which protocols to use, which SOPs to use. And so that's what this webpage does, and it's laid out really nice and neat, and it has plenty of information on it as well. And so what we're going to do is kind of talk about where individual products may fall and how to achieve that testing. So in the umbrella of a residual claim, we have 2 major categories, with one of them being subdivided up into 2 slightly smaller categories. So the first category we have are the residual disinfectants. So these are the disinfectants that are applied to a surface that continue to provide residual efficacy. These residual disinfectants, they meet the standards for an EPA standard for a disinfectant. So they have passed the evaluation of the germicidal spray or the used dilution. Unfortunately, towelettes don't really fall into this, but that's something that can be discussed with the EPA for developing a method for that. And then we have the supplemental residual antimicrobial products. So the keyword here being supplemental. These residual antimicrobial products are used in conjunction with a residual disinfectant to produce longer times of efficacy with these products. So these are coatings, films, solid surfaces like a copper handlebar, and paints. So those are just a couple examples. And the key here is these products do not meet the EPA standards for disinfectants. So these products have not gone through germicidal spray testing or used dilution testing or non-food contact testing. So what we do is we use a machine that looks a lot like this. This is the Gardco washability and wear tester machine. So this machine has that boat that's right in the center there on the test platform, and it slides back and forth for a set number of passages. And so what the EPA has really done here is they've used this machine and they've calibrated the number of passages and the length of time it takes for a passage. They've calibrated the sponges that you use, the cloth supplements that you use for durability testing. The weigh boat weighs a certain amount. So everything is calibrated so that when this test is performed here at our Bozeman site or down at Nelson Labs in Salt Lake City or over at our Fairfield site, everyone is performing the test the exact same. Everyone is wearing the product the exact same. And so then for 2 of the tests, you actually use additional chemicals to represent real life scenarios where people are using these products that are previously approved by the EPA, and then using the supplemental, when used in tandem, we're proving that there's no adverse effects. So this is a quick video of kind of what this machine looks like and works like. And so each of those passages is timed back and forth, and the weight is the same. It's not the same machine. I couldn't unfortunately find that, but I felt this was a really good representation of what's happening. So we'll go into a little bit more specifics here and talk about the residual disinfectants. So just keeping in mind that these are products that have previously met the disinfectant efficacy standards implemented per the 810 OCSBP 810-2200 guidelines. So when we're looking at these, we're looking at using the EPA's interim residual self-sanitation protocol, also known as protocol number 01-1A. And again, these must have previously met non-residual disinfectant claims. Typically, this is going to be tested against Pseudomonas and Staphylococcus for, say, a hospital claim for a used solution or a germicidal spray. The carriers that are tested on for these are 1 inch by 1 inch surfaces, hard non-porous surfaces, stainless steel, as an example. 2 lots of the disinfectant are tested at the lower certified limit. So that's kind of a little different from normal EPA supplemental type testing, whereas for a used solution, if you're adding a supplemental claim or the germicidal spray, something like that, you are allowed to test it at the nominal concentration. So what we're looking at here is supporting a 24-hour residual claim. So that's what this method allows to go on the label, is for up to 24 hours, this product continuously reduces the antimicrobial the populations of the microbes on the surface. If you're looking for a longer or a shorter claim, you should discuss with the EPA what that looks like. Maybe it's a slight alteration to the method, or maybe it's a completely different method. That's up to the EPA to decide. So, and then again, this is not intended to address residual claims for towelette products. And so I would assume that the EPA will have an alternate method or modifications that can be used for those. So for a bactericidal claim, we're looking at testing Pseudomonas and Staph. Again, these are the bugs that are used for hospital claims, and that's what they're targeting as the most difficult to kill of the bactericidal in the EPA's playbook. So these require a greater than or equal to 5 log reduction in less than or equal to 10 minutes. And I believe there's a plus 5 seconds allowed on there for recovery time. So that's when compared to the parallel control. So that'll be the same carrier surface, but that hasn't been treated. And I'll show what the treatment looks like in a sec here. And then if you've passed with the Pseudomonas and the Staph, you can actually bridge the data from previous non-residual disinfectant claims for additional bacteria that can be bridged onto this label as well, and the testing of those is not required per the EPA. So, and then on the virus side of things, what you're actually looking for is the most difficult to kill virus on the product label is all that has to be tested. And the EPA has guidance on that, and any of our virologists here can help decide that as well. So the viruses are only looking for a greater than or equal to 3 log reduction in that same 10-minute time point when compared to the parallel controls. And then it does require a little additional testing with the ASTM E1053 to ensure that the viral recovery is sufficient. Claims for additional viruses can also be bridged from the non-residual virucidal data. So if you've already tested them in the used solution or a germicidal spray, that data can be applied, and testing of the additional viruses is not necessary. So this is kind of what the general testing flow looks like for this protocol number 01-1A. So it's about a 24-hour product application and wear cycle. So this all happens over the course of 2 days about. So you start with the initial inoculation of your test organism, and then you apply the test product in a way that's consistent with the application of the test product as decided by the client. So you start with 1 wear cycle with a dry cloth, and that's a very specific cloth that's specified in the protocol. And then you go through the process, which is one forward pass, one backward pass, and then you re-inoculate with the test organism and you let it dry. And then you do it with a moist cloth. And so that moist cloth is just saturated with a couple spritzes of water, and then that just goes over and back. And then you re-inoculate and dry again, over and back, this time with a new dry cloth. And then you re-inoculate, and that's the end of your first day. Coming in the next day, you just start the process, you continue the process with additional wears until 12 wear cycles are completed. And then within at least 24 hours of application of the test product, you start the sanitizer test. And so once all that's done, the residual disinfectant claim can be made. And then moving on to supplemental residual antimicrobial products, these are the coatings, films, and the fixed solids that are impregnated into a surface, perhaps. So in reminder, these do not meet disinfection efficacy requirements for the EPA. So what we're looking at here is for the surface coatings and films, we're looking at testing using the new EPA SOP MB40. And so this effectiveness is determined up to several weeks. And then the fixed solids and surfaces are evaluated using the SOP MB41, and this effectiveness is determined up to several years even. So here's where we're getting away from that residual disinfectant, a product that's potentially applied every single day. These are products that are built into a door or some sort of film that's put on, let's say, a hand railing to reduce. So those are intended to last for much longer than a daily application. So again, for the bactericidal claim, we're looking at Pseudomonas and Staphylococcus again. Virucidal, again, the most difficult to kill, that's going to be claimed on the label. So these are again tested on hard non-porous surfaces in 1 by 1 inch sizes, which are stainless steel. And then coming back from the residual disinfection, we're actually only looking for a 3 log here. Because these products don't meet that disinfection efficacy requirement by the EPA, they're not required to have that 5 log reduction. So they only need to show the 3 log reduction when compared to the parallel controls. And again, 2 lots of the disinfectant or the film at the lower certified limit in accordance with the OCSPP 810 guidelines. And here's something slightly unique to these. When you're looking to make a viral claim, you must first show bactericidal efficacy or concurrently with the viral claim. So you can't test your virus first and then submit that to the EPA for approval and then come back and do bactericidal, or even not do bactericidal. You must do the bactericidal concurrently with or before submitting a viral claim. So when we're looking at this method for the MB40 SOP in a little bit more detail, what we're looking at is performing it on those hard non-porous 1 inch by 1 inch surfaces. And the EPA isn't necessarily opposed to testing on porous materials, but it is something that should be discussed with the EPA prior to initiation of the evaluation. So again, testing with the Pseudomonas and staph, really looking for that hospital claim meeting those same bacteria that are required for the disinfection claims. And again, the hardest to kill virus. The one difference here between the residual disinfection and the supplementals is there is no bridging of data. So what you're looking at is every additional microorganism that you want to add to the label also needs to be evaluated. However, it doesn't require the durability assessment. So it's just the application of the product and testing after the set time. So with the MB40, this is the one that supports a claim of up to several weeks. The one that's specifically talked about is for a 1-week claim, and longer claims should be discussed with the EPA prior to testing to determine how they are recommending to meet that. So this testing consists of 10 alternating chemical and drying cycles. And so these chemicals are really in-world real use disinfectants. These disinfectants are up to the customer to decide or the laboratory. However, they must come from the EPA list of approved hospital disinfectants. So the first one is a 2,000 part per million sodium hypochlorite solution that's mixed up in the lab and used as a bleach type solution. And then the second one is a hydrogen peroxide and peracetic acid product that is currently on the market with a hospital registration. And then the third one is a quat product that is labeled as a one-step cleaner. So that means it's a product that's been tested with a soil load to ensure that it is efficacious, even with an organic soil load, potentially inhibiting the product. So again, there's 2 lots of product need to be tested, possibly at the LCL. The method and the guidance don't really align on what they're saying in this, and that would just be something to clarify with the EPA. The SOP says that it's just two products at market level, and then the guidance says at the LCL. So it's really just not clear there, but it's something that can easily be determined with an EPA consultant. And again, as these are supplemental, they don't require the 5 log, but they do require the 3 log reduction when compared to the parallel controls, with a contact time of 1 to 2 hours. So that's a hold time of the microorganism on the surface for 1 to 2 hours. The EPA doesn't recommend times less than 1 hour or more than 2 hours, as that's not really a real world application of the product. And there's a little bit more later that'll talk about that. And so what we're looking at, so this is just the general test overflow. So what you're seeing in the top left there is the testing surface of that durability wear tester. And then each of the silver squares are the control slides, and each of the orange squares are the test slides. And so those are secured to that surface with magnets, say, or a template that can be fashioned for it. And so when I say this happened between alternating wet and dry components, that's kind of what we see in table 2 there. This is 1 cycle, which is 4 passes. So that's 1 pass forward and back, and 1 pass forward and back. So 4 single passes over it. And then that wet component, so that's treated with each of those chemicals that were discussed. And you can kind of see that down on the bottom one there. Treatment A would be the sodium hypochlorite. Treatment B would be the hydrogen peroxide and peracetic acid. Treatment C would be the quat. And treatment D is just a dry abrasion control. So what happens is it flows through this 5 different times. And so we have our totally untreated controls and our treated control that are not run through the machine. And then we have our abraded test and control, which are run through the machine. They're run through the dry and wet alternating cycles for a total of 5 times, total of 10 times for the dry abrasion, and then they're evaluated. So the evaluations need to take place within 7 days of inoculation of the product. So the product is applied at the beginning and dried, and then it's run through the process. This one, unlike the protocol number 01-1A, there is no product, no microorganism applied in the test until the treatment cycles have concluded, until the wear cycles are done. So once everything has gone through the 5 cycles, then the microorganism is inoculated onto the surface of the slide, exposed for the 1 to 2 hours, whatever the contact time is chosen as, and then neutralized and recovered for quantitative data. So now looking at the fixed solids and surfaces, the copper, ingrained in a handle or a railing, we're looking at using the SOP MB41. So this one is, again, performed on 1 inch by 1 inch samples of the product. So these are supplied by the client. And then for any non-copper surfaces or paints, the EPA does recommend consulting with them just to make sure that everything can move smoothly through the process and not be tied up in stuff that could be avoided early on. Again, we're looking at Pseudomonas and staph and the hardest to kill virus. The difference here is that the testing consists of 150 abrasion chemical rinse cycles. So we'll go through that one as well. So there's a lot of preparation for this evaluation that goes on. These abrasion cycles take place over 5 weeks. So you can go for 5 weeks and then test on the 6th week. So duration claims are not necessary. You don't need to say that it reduces for 2 years, 1 year, 6 weeks kind of thing. And the chemical cycles are the same 3 chemicals chosen from the EPA hospital disinfectant list. So here it is, 2 production lots of the product should be evaluated. So those are products created on different days or different machines to make sure that the production of the product in its entirety is efficacious. And again, this just only requires a 3 log reduction when compared to parallel stainless steel controls, again, with that contact time of 1 to 2 hours. And much like the previous one, the MB40 testing, there is, again, no bridging of data. So any additional microorganisms need to also be tested, but again, avoiding that durability assessment. So it's just does your product still work against these? So this one's a little bit more straightforward, but it's just more drawn out. So it's the same test surface placement, the same slide placement as the previous evaluation. And then this little diagram below that shows what happens for each of those chemicals. So you have your 5 copper type products and your 3 stainless steel controls. They go through the abrasion cycle. They get treated with the disinfectant. They get rinsed with deionized water, and then they're let dry for 30 minutes. And then you go back and do it again. And so on each day, that's repeated 5 times. And so for a total of 150 treatments over 5 weeks. And then following all the treatments, then you take it and you inoculate it just like before, expose it, neutralize it, sample it, done. So please discuss any label claims with the EPA, as their guidance doesn't exactly include every single scenario. I can't speak for the EPA on how they would feel about anything. So if you have any specific questions with a claim, a label claim, you do need to speak with the EPA. However, this is the information that was provided in the guidance documents. So for that residual disinfectant claim, you are eligible for inclusion into the EPA list Q, and that can be looked up on their website. And then additionally, this product, the residual disinfectants, if they pass the evaluation, they've already passed the disinfection evaluations, they can just be used as standalone disinfectants and do not require any supplemental notation or supplemental wording that additional products are needed to be used with this product. But when you get down to the actual supplemental residual antimicrobial products, there's a little bit more going on here and a little bit more clarity needed with the label. So the products clearly need to be labeled as supplemental to standalone disinfectants. They can also be included in the list N appendix. So the list N appendix is just an appendix that lists products that are supplemental to previously approved standalone disinfectants. Due to the longer contact time and the lower reduction required, these do not qualify as residual disinfectants, as we discussed earlier. And the label must clearly state that the product does not meet the EPA's efficacy standards as a standalone disinfectant. So you need to be really careful with that label to ensure that you're not making it appear as though the product is a standalone disinfectant. So, and then looking at the fixed solid surfaces, we're looking at the same things. However, it should be labeled as supplemental residual antimicrobial surfaces, not as a supplement to. So it's you're focusing on it's a surface that has some antimicrobial aspect to it and not a disinfectant or supplemental to a disinfectant. So again, this product can be included on list N appendix. So it's again in the back of list N, the product would be included to be used with any other standalone disinfectants. And again, this does not qualify as a residual disinfectant due to the longer contact time and lower reduction required. And it also needs to be labeled that, very clearly labeled, that it does not meet the EPA's efficacy standards as a standalone disinfectant. So with that, that's the conclusion of the webinar. Does anyone have any questions?
Thanks so much, Grady, for your insights. We learned a lot of information today, and as you just mentioned, we do have a couple questions that would be great if you could address now, and I'll start going down the list. The first of which is, does the active ingredient matter when making residual claims with the EPA?
Yes. So for the claims for the supplemental, they do focus on copper as the ingrained antimicrobial. However, they just ask that you talk to them more if it's a non-copper based product. They just want to make sure that everything is being handled correctly, and they just don't have enough information, data yet to do anything beyond that. Now, for residual disinfectants, yes and no. So what the residual disinfectants have already shown is that they are effective at the 10-minute time point as required by the used dilution or the germicidal spray. So the active ingredient doesn't necessarily matter at that point. What we're doing in this evaluation using the protocol 011A is showing that that product, that active, still has efficacy after a 24-hour wear cycle. So does the active actually matter? Not necessarily, but definitely discuss with the EPA if it's something unique or novel.
Thank you so much for that well thought out answer. Second question is, does a coating need to be registered with the EPA before testing residual claims?
I'm not entirely sure on that one. That would definitely be a question for the EPA consultant that the client is working with. But it kind of seems that this process is how you go about developing that claim with the EPA.
Fantastic, thank you. And we have time for one more, and it's, can you make residual claims for food contact surfaces?
Currently, no. There was no indication in the guidance or anything I've read to where any of this applies to a food contact surface. So food contact surfaces are, because of the sensitivity of the area on food prep stations, in food manufacturing plants, relying on a residual disinfection process that takes up to 1 to 2 hours to be efficacious is not entirely what is hoped for in these types of products.
Fantastic. And if we didn't get to your question today, Grady will be happy to address it individually after we conclude. And as you can see here, please reach out at experts@nelsonlabs.com to ask any of those specific questions. Thank you, Grady, for that thorough and informative presentation. I'd like to thank you, our presenter today, for your insights. And I'd also like to thank you, the viewer, for attending this session. I hope you found it valuable and informative. And please be sure to visit us at nelsonlabs.com for more on-demand and live webinars like this. And I hope you have a wonderful day.